Hi, I'm Kim Snowden. I'm a plant scientist who specialises in genetics, and I lead the team who are using the latest tools in gene editing so that we can alter plants to improve them for our future. I'm going to take you through into the office and the lab, and you can meet the team and we will show you the steps that we use to edit a plant.
When we've decided that we need a new plant with a new trait, the first thing we need to do is sift through all the information that we have, both internally here and what's been produced around the world. Then we bring all that knowledge together. Mostly it's DNA at this point, and it goes into our computers as sequence files. The files are really large, but because of the tools we use, we can narrow in on just exactly the right place in the genome. The place in the genome that we can edit to change the trait that we're most interested in. It can be quite a complicated process, but we have tools that help us do it reliably and predictably.
So once we've planned the gene editing on the computer, it's time to build a construct that we'll use in real life in the lab. So gene editing takes lots of different components, and we combine them together into a construct and insert that into bacteria to make lots of copies. Then we use techniques like PCR to check that the bacteria has the correct construct, so we know which bacteria to use going forward.
We use agrobacterium to deliver CRISPR-Cas gene editing constructs to plants. We start with a liquid culture of bacteria that carries the constructs. When it's ready, we take the young leaf from tissue culture, we prepare them and apply the bacterial solution directly onto them. We let them sit together for about 30 minutes. Next, we place them onto the growth medium and into the dark for about 2 days, after which we bring them into the light and onto the selection media so that we can eliminate the bacteria from the plant and select the plants that have taken up the construct.
So once we take the plate out of the incubator, we will select the positive tissue and then transfer them into new media to encourage them to grow into a planet. And then after that, we will transfer the little planet into the rooting media to encourage them to grow roots. Once we have the roots, we will take them down to the glasshouse and transfer them onto soil. Depends on the species, this process can take 6 to 12 months.
So I'll get down to the greenhouse and I'll pick a plant and I'll take a leaf off it and bring it up to the lab, freeze it in liquid nitrogen to break open all the cells and release the DNA. And then we'll look at that DNA using a number of different tests in the lab, including this fancy digital droplet PCR machine. And then we'll work out whether we've got exactly the right edits and exactly the right number of copies of the genes in the plant.
So once we've done all that work in the lab, we will know whether which plants have edits and which ones don't. And we can go to the plants in the glasshouse and compare those plants and look for changes to their growth and development. So as an example, I have some plants behind me, they're tamarillos, and we have some that are not edited and they are tall and no branches. And then we have edited plants and they are a bit shorter and they have increased numbers of branches. So that will overall affect how we can manage those plants. We can grow them more crowded together and increase the yield. Today we've just shown you a small snippet of the months and sometimes years of work that goes into producing gene-edited plants. We're really passionate about understanding the biology of plants, and that is really needed for us to be able to grow plants for our future.